On this podcast, you'll find interviews with high-performing, successful individuals in life sciences. On a weekly basis, we cover their proven methods, principles, strategies, and mindsets to implement new technologies that scale to meet the needs of people in our world.
Hello and welcome to Life Science Success Podcast. My name is Don, and for those of you who don't know me, I'm a consultant in life sciences. I help companies manage complexity and increase performance. This week, I'm joined by Raymond Peterson. He's the CEO of DNA Analytics, and we'll be discussing the use of AI in establishing DNA targets. We also are going to have an interesting conversation, it seems, in therapy for viruses with honeybees. So Raymond has some involvement there as well. And so welcome, Raymond.
Well, thank you, Don. I appreciate being here. Thank you for having me on.
Yeah, thanks so much. So can you tell the listeners just a little bit about yourself?
Sure. So I know sometimes you ask people, how they come to science. And so I feel like I came to science maybe a little bit later than others. I was in my mid-20s and decided to go back to graduate school in science and was interested in answering the question of sort of like where as humans we came from, and was interested in more of a genetic perspective. And when I was in graduate school, met a professor that was very much involved in genes. And at the time, it was called population genetics. Today, it's called maybe bioinformatics, but it's the mathematical modeling of genes as they descend through populations and modeling evolution. And so I got really enamored of that. Just really loved the technology and the theory of it. And then have established a career with that. I ended up getting my PhD in genetics through the National Institutes of Health as a fellow, and then went to the National Cancer Institute as a researcher. And when I was there as the researcher and we were looking at genetic diagnostic assays, given my, modeling background and computer software expertise, I realized that people were struggling with their assay designs, and so left the Cancer Institute, took some of the technology with me, and established DNA Analytics, where we use artificial intelligence to identify the best match of genetic probes for diagnostic gene therapy and gene vaccines, and it helps speed to market these therapies and diagnostics by a matter of months and years.
So are most of the companies that you're working with that DNA Analytics, are they newer companies? Are they, more established, or kind of a mix?
It's a bit of a mix. We do tend to work with more startups because the way that our algorithms work is that it helps young startups save time and money on the laboratory phase. But we do work with some of the larger biotech companies in the tool space, the tool companies, your sort of like Bio-Rad type companies.
So even more in the, I guess whenever I think of Bio-Rad, I'm thinking like more of a diagnostics kind of company as well.
Well, that's right. So it depends on the company. Some companies develop their own assays in-house, and when they do, they have a need for our software because, again, it helps scientists develop these assays. But then for the tool companies, they develop assays themselves, and then, of course, they sell the tools to others. And so you might see our software that drives an ordering site on a biotech tool company, where you go in and say, well, here's my gene. I really want the best assay for this gene, and our software will operate on that and return back to you the assay. And then the components of that will then get sent right on to manufacturing through the tool company, and the reagents will then be manufactured and shipped to you.
I do love the fact that our science is at such a point that you could say, here's my target, can you establish a diagnostic that more or less, results in that? And for sure, the work that you've done, in the bioinformatics pipeline in that space, for sure, has to help, companies as well.
Yes, well, it sure does. And so what we focus on primarily is the really difficult assay context. So probably, as you know, in the human genome, there's something like 4 billion nucleotides. That makes quite a design problem because there's only the four letters A, G, C, and T. And so there's only a limited number of contexts that you can design to. And what our algorithms do is we determine to a more accurately and precisely the match strength of what are called these oligonucleotides. Probably many of the audience here is familiar with oligonucleotides, probably a lot of scientists, but they're the short sections of genes that get manufactured that allow you to target that individual gene and to not target other areas of the genome. And so our software identifies the best locations for those oligonucleotides, and in particular, using engineered nucleotides that scientists have developed to sort of aid this process along. One of them is like locked nucleic acids, which is widely used in the industry. And the benefit of a chemical like that, as compared to natural DNA or RNA, is that it helps increase the match strength. And then when so diagnostics, they typically want to go into the body. But if you're doing, say, gene therapy with an oligonucleotide, the LNA would help resist degradation in the body. And so it has another benefit there as well.
Very good. And then also in your career history, I saw that you work with Granite Point Ventures. Can you tell us a little bit about them and who do they typically invest in?
Sure. So good question. That's right. So with Granite Point, I learned over the past, say, 15 years as I've developed DNA analytics that myself and the sort of network that I've developed are really good at developing and bringing technologies out of academia and out of universities and into the marketplace. There's kind of a real gap there where you'll have something that looks really pretty good to the researcher, but then with our experience in diagnostics, we know where that technology really needs to get to before it's commercially viable, and so that's where we fill that gap in. For example, one area is you had mentioned before the honeybees. So where I live here in Maryland, the USDA has a research facility. It's actually the largest research facility in the world for agriculture, funded by a government. And there's the Bee Research Lab. And so one of my population genetic friends who works in insects rather than humans, like mostly I've done, said one day, "Hey, well, it's we've got this problem with honeybees. They're dying. They're not going extinct because we're able to replace them. This is costing the industry $250 to $300 million a year to replace the honeybees because they're dying of disease." I said, "Well, let's take a look at this, and sure, I'll see what we can do." And so we actually did take and looked at one of the diseases that is the primary driver of killing the honeybees, which is a virus. It's called deformed wing virus, and it prevents the bees from developing properly. And so we've raised, through sort of my experience with DNA analytics and sort of knowing how to kind of work through the system, we've raised about $1 million to develop a therapy for the deformed wing virus. And there we're partnering with another artificial intelligence company called Atomwise, which is in San Francisco. The approach that we're taking with the honeybees is actually not genetic, but it's a small molecule chemical. And so Atomwise does their artificial intelligence to suggest which of these chemicals out of millions and millions in their chemical, virtual databases might interact with the virus. And so we ran that virtual screen, which resulted in under 100 chemicals for us to actually physically screen in the library, and in the laboratory. And so we are now, that project is kind of transitioning between discovery and hit to lead using the pharmaceutical nomenclature, where a hit is something that looks promising in the laboratory, and then a lead is something that actually has more evidence that it's inhibiting the growth of the virus.
Very good. Yeah, I mean, I definitely important work, for sure. And it'd be great to, to see that some of the bees could be saved, as well. I know that pesticides have done an awful lot of damage, over the number of years, as well as getting rid of, a lot of the things that the bees need to just support them. So in general,
Yes.
I look forward to the good work there.
Well, thank you. Yeah, so it's the work for the honeybees tends to be a little bit easier to explain than the work for DNA analytics, because for DNA analytics, it's really sort of really right at the scientist level, whereas people really well understand viruses. Well, take the last 2 or 3 years, and one of the not benefits, but one of the consequences of the pandemic has been that many people better understand what biological scientists are doing on a daily basis, and we've all become amateur virologists, which is, I think, a good thing.
Yeah. Yeah, I mean, I know I've had even, people in my neighborhood take a dramatic interest in what I do, on a daily basis. And some of them, I think, even come and listen in on the podcast that we have to just try and better understand what are people working on in this space.
Yes.
So can you tell us a little bit more about DNA analytics and what is the focus of DNA analytics, more specifically?
Yes, certainly. So as I had mentioned before, that there are these 4 billion chemical letters in the human genome. Other genomes have more or less, depending. And so what we did, which was very highly unusual, is that we developed statistical algorithms that extended algorithms that already exist in the field, in particular for these modified chemistries. And so we then went to one of the large oligo manufacturers and said, well, geez, we need several thousand of these oligonucleotides to measure in the laboratory to determine their physical properties, from which we could statistically train our algorithms. And so we spent probably about 5 years in the laboratory measuring these actual experiments. And from the experiments, we were able to extract the statistical predictions so that we could then make predictions on any novel sequence. And the way that these are used is that it probably, as we all know, you maybe look at a gene, and well, actually, let's take a really great use case, which is genetic variation, and compare the two. So, one application is, say, gene expression, another is genetic variation. For gene expression, you might have a whole gene. It's maybe 2,000 letters long, 4,000 letters long. And if all you want to know is this, gene expressed or not, you've got that whole 4,000 letters that you can work with to design an assay, which means that you can usually find maybe a amenable sequence in that area. When you go to a genetic variation, you've got a lesion or a change in one base, maybe in one very specific location. You have no choice but to make an assay exactly right there. But that sequence may not be amenable to making an assay. And so what our algorithms are able to do is to look at the biophysical properties of the nucleotides, see how strands fold and how they match and the strength. And we're able to determine, typically a clean assay where you're able to get good signal from your the gene that you're looking for, this genetic variation that you're looking for, but also don't detect signal from other genes, especially, say, closely related genes.
Very good. And then are most of the companies that you're working with, are they targeting drugs around cancer therapies or other things as well?
So it is some on cancer. Often it's other things. And if it is cancer, where we really shine is in the rare diseases. And the reason is that for big diseases like the popular cancers, if you will, there's such a huge market out there that that the market demand is there to make to spend a lot of money in developing assays and therapies. Whereas for the rare diseases, that's not the case. And so our technology, by dramatically reducing the cost that it takes to develop and the time, either a diagnostic or a therapy, we can really target those rare genetic diseases or just rare diseases in general, and be able to come up with a therapy that is justifiable based on the market size of that disease.
So I'm going to take a little bit of an aside, but then I'll come back to kind of this topic of your genetic assays, as well as really what we're seeing with regards to personalized diagnostics. But recently in the news, one of the things that came about was Google and this idea that they have an AI chatbot that became sentient was kind of the idea. Whenever
Yes.
I think about, I mean, and this kind of goes to one of the key problems, I think, whenever you talk about artificial intelligence. It's like, in this case, we're talking about something that's helping in my mind with like kind of visualizing where the needles are in the haystack, right? And
Yes,
it's sort of,
exactly.
pinpointing versus like independently thinking and creating something that could be scary. I mean, I guess could you maybe provide any thoughts that you might have with regards to what happened with Google, and then also I'll have kind of a follow-up question with regards to kind of the future of this segment of science as
Sure.
well.
Yeah, so that's a very interesting case. Of course, Google has denied that their algorithms are sentient.
Right.
um, yes, even 20 years ago, when DARPA was funding self-driving cars, which have now turned into a lot of the technologies that are going into Teslas and all the other cars. A friend of mine came up to me and said, "Geez, are you concerned about this? That the AI is going to take and all the robots are going to take over the world?" And my answer today is about the same then, which is that there are concerns, but it does help us in many ways, being able to filter through a lot of noisy data that we're not able to ourselves sort of understand and pre-build a model that addresses all of the variation and all the causes that goes into the sort of the outcome that you're trying to model. Because traditional statistics, you have to build the model first and then see if it fits your data. Whereas in a lot of ways, AI is going in reverse, where it's taking the data and in the best cases, the data then are able to speak, and we're able to then have models fall out of the data. I think that that chatbot from Google probably can be distinguished from a real person. It just depends on perhaps which subject you're talking about, and so I think that there is obviously a lot of value to these artificial intelligence algorithms. So often some misunderstandings. In our particular work, we're in what's called machine learning, which is a little bit different than what some of these fuzzy networks do of where we had our own models and we had science that was and prior models that were driving our work, and so we were able to build the models in advance and then know which data we wanted to collect to train those models. Whereas the chatbot is kind of working in reverse, where it's you're telling it, gee, these are maybe here, here, and here is the things that I want to know, and then it goes and finds things that are related to that. Um, I don't know. In some ways it's very disturbing, but on the other hand, I'm pretty happy when I'm in a rush and I get in the car and Google tells me, "Oh, you have an appointment in 15 minutes that you're late for. Here's your directions. Just press here." I'm like, "Oh,
Yeah,
thanks."
here's a better route, right? Yeah, exactly.
Right.
Yeah. Yeah, and so just really quickly, I'll add this to to the window here as well. So this is Brian Barsher. Came up and said that you have great insights, and thank you for putting those out there as well. So kudos.
Oh, great.
Um,
Well, thank you. Brian's a really
yeah.
good guy.
So also, I guess the follow on question that I would have to this as well is that. What do you think is kind of the next thing in our industry? What do you think we'll see, come about next with regards to either artificial intelligence, or you could take this, down the liquid biopsy or diagnostic space as well. But I'll leave that question and answer, more or less up to you in terms of
Sure.
how you answer it.
Right. So what I'm seeing and what I believe is that these algorithms are going to, and approaches are going to make operations more efficient. That in clinical diagnostic labs, there's obviously a very issue of being able to have a very quick turnaround time of samples that come into the report going back to the physician. And diagnostic labs are, to my view, very complicated manufacturing processes. And when we get to the end of the assay process and that report can't go out because something's gone wrong, we've got to go back and restart the process from the beginning. And clinical labs are always struggling with keeping their quality assurance proper at each step along the way. And I think that this is one area where the artificial intelligence will really help us, because it's really going to help us identify problems that are starting to arise before they turn into failed clinical diagnoses.
Yeah, it's one. Kind of the last part of your answer there is something that I oftentimes refer people back to whenever they ask, do you think that artificial intelligence will take over your doctor's job? And I've answered this publicly before on other people's podcasts, that essentially, look, I mean, if your doctor is the one that follows a checklist and more or less has like zero empathy, I think where we're at today, then yeah, I think artificial intelligence could do that pretty well. But when it comes to applying scientific theory or having to be creative in terms of solving a particular problem, right now that's not really where we're at with a lot of these things. Where that idea of being creative about solving a problem that doesn't actually end in something catastrophic isn't something that we're able to do today. And so I do look forward to the advancement, because at the same time, with 50 to 60 new drugs being released every single year, it's got to be hard to keep up as well. So having things that help us decision better seems like a good thing for healthcare, at least today.
Oh yes, for sure. And speaking of the drugs, there are drug interaction databases, and so being able to flag whether the physician is perhaps prescribing something that the patient is already taking that might have an interaction that they weren't aware of is a great use case. I think that there are obviously many companies out there working on decision support systems, and I think that that's the role that it's going to play. Isn't it so much being a telemedicine doctor that you're just talking to the computer and then it's going to spit out a prescription to your pharmacy? I think it's going to help the doctors make better decisions and help lead them through the questions that they're able to ask. Perhaps as actually I think about this time of year, isn't it, that the residents and turnover in the hospitals, and you can tell that because right now somebody's new and they're maybe the first time that they're actually interviewing patients, and so then they ask a lot of questions, but then you can tell somebody who's more experienced because they kind of get through those questions a lot faster, a lot easier, because they have more experience and can guide that.
Yeah, absolutely. So there are 3 questions, Raymond, that I like to ask every guest. What inspires you?
So what inspires me is, I think, the young generations, which I hate to say I'm even in the position to say it that way, but a lot of people that are coming through college and working through life, I think that they've been socialized in very different ways than both I was, probably you were, our parents' generation. And I think there's just a lot of real positive thinking and motivations going on. And so I'm really looking forward, actually, for perhaps the current generation of leaders, turning the reins over to younger generations and letting them have a shot at solving some of the problems that we weren't able to solve.
Yeah, it's funny that you mentioned that one. So I recently was telling somebody this story. I went through a few years ago now. I went through and I interviewed some interns, and I was so mystified by these resumes of the interns. I mean, they were mentoring people, they were tutoring people, they were working for charities, they had involvement in STEM. I mean, it was
Yes.
crazy, all of the stuff that they had. And they grew up, in a completely diverse environment,
Yeah.
unlike where I started out. And so I just kind of, looked at it and I was like, to be honest, I mean, I was glad at that point in time that I wasn't competing against them in the job market. So, but obviously all of them are out now. And now my question that I more or less had asked of this individual was, I wonder what the future will hold, right? Is it that all of those things tie to, a lot broader perspective and yield something great, which is what I would believe, or are there other challenges that they face that I'm just, unaware of as well? So,
Yes.
yeah.
Well, that's so true. Well, and their challenges are going to be, some the same and some different than ours.
Right. Yeah, exactly. And my follow-up question is, what concerns you?
So what concerns me is uh, sort of the integration of different aspects of our society. So I know that this is perhaps not so much life science, but when I was coming out of college or even high school, had somebody told me, "Oh, you have a 2-year Army commitment, or you have everybody coming out of high school has to serve, no matter where you are, and you just get mixed together." I would have been like, "I'm not going to do that." Or a Teach for America. It's a required thing. I wouldn't want to do that. But today, I'm thinking that would be a really, really great thing for our country. They exist, but something like, the Peace Corps, but internally, we need people in cities going out to the country and vice versa. So that and as young people, so we just interact more and start forming a basis of sort of mutual understanding and seeing that, we're not really that different. You might drive a Ford truck. I drive a Ford sedan. They're both Fords.
Yeah, this idea of having perspective, but then also be able to utilize that perspective for greater, maybe it's greater discourse, maybe it's better challenging your own thought processes, but for sure, a lot of these service programs for sure would be great to have as well. And I have 2 daughters who have served as well, and
Oh,
I'm
great.
proud of the fact that they both decided to go to the military first, and then
Ah,
they've decided, one of them decided to leave the military, and the second one decided that she's going to make it her career. So
oh,
yeah.
nice.
And what excites you?
So what excites me is the technology that's being developed today, both in the life sciences and broader. I read something several years ago about how the amount of information that we have to process on a daily basis, say just like one edition of a newspaper, is about the amount of information that somebody would have to have been exposed in their entire life in the 18th, 19th century. It's just
Right.
crazy how we're inundated with information. But there's just so much going on between making safer cars, transportation options, carbon sequestration, and then of course in life sciences, we're really making a lot of progress in understanding both deconstructing the body and how the different pieces fit, but then also putting it all back together and seeing how the whole is greater than the parts, but addressing that whole as well. And we're cancer was maybe stalled out for about 30 years, where the rates of cures were not very different. But then now we've got immunotherapy, and there was actually an article very recently, a very small sample, but I think they were able to put the cancer into remission for all the patients that were in the trial. Maybe it was only all 12 of them, but that's really a positive thing. And so I think somebody once asked me, it was an uncle, says, "Do you think we're ever going to cure cancer?" And at the time, thought, well, and I still have the same answer. It's like, well, no, because cancer sort of happens and there's nothing we can do to prevent the mechanism from it happening. But can we cure it in the sense of once it does happen, can we detect it early and then have a treatment for it? Yes, I think we can.
Yeah, I feel like there's all these guys that are working on epigenetics. There's, people, very focused on the microbiome and all of the, sort of possible indicators, ahead of time, in addition to, just all of the science around kind of the back end of cancer as well. And
Yes.
it definitely is an exciting time, from a scientific standpoint. And then, to think about the fact that, who knows what CRISPR will eventually do. Who knows what, some of the new, even, CAR T therapies or CAR M therapies that are out, what they'll
Yes.
eventually do. I mean, it's just an amazing time. Yeah, I agree with you on that front as well.
It is. And then once we do get these therapies, of course, the challenge is developing the technologies so that they're, affordable and easy and able to be distributed, sort of equitably around the country and to different people, different backgrounds.
Yep. That's for sure is one of the bigger challenges, I think, in the CAR therapies to try and solve right now is how do we extract, what you have and then kind of deliver it back, in an equitable, amount of time as well. So, well,
Yeah.
Raymond Peterson, thank you so much for being a guest today on the Life Science Success Podcast. I greatly appreciate you being here.
Well, great. Well, thank you for having me. I've really enjoyed it.
Thank you for listening to Life Science Success. For complete details about this podcast, including show notes, how to get in touch with guests, and more episodes, please visit www.lifesciencesuccess.com. If there's someone you'd like for us to invite to the show as a guest, please let me know by sending me a message at the podcast website. Please click subscribe on your favorite podcast app, share the podcast, or tell a friend about it, and last but not least, rate the podcast. Thank you again.